This protocol is the result of evaluating key aspects of circRNA library preparation, such as kit type, RNase R treatment, input amounts, and their impact on circRNA detection. It enables optimal circRNA detection and provides data on adjustable parameters depending on the needs of the user. Identifying circRNAs in different sample types will help us to better understand the distribution of their expression and sets the stage for delineating the functional role of these molecules. This method can be applied to any total RNA sample. It is important to keep RNA samples on ice prior to beginning the protocol, assess the RIN and DV200 values on the Agilent bioanalyzer or tape station before the prep, and follow appropriate procedures when working with RNA.
Begin by diluting total RNA to 4 micrograms in 39 microliters of RNase-free water and pipetting it up and down to mix. In a separate tube, use 1x RNase R buffer to dilute the RNase R to a working concentration of 2 units per microliter. Pipette 39 microliters of total RNA and 5 microliters of 10x RNase R reaction buffer into a 1.5 microliter reaction tube and mix. Add 6 microliters of the diluted RNase R. Then adjust the pipette to full reaction volume and mix the solution by pipetting up and down 10 times. Place the tube in a 37°C water bath for 10 minutes, making sure that the full reaction volume is immersed in the water bath. Then place the tube on ice and immediately proceed with RNA cleanup and concentration.